Hello everyone and welcome back for more biotechnology. So in this chapter we are going to start rediscussing a lot of the DNA, recombinant, genetic engineering topics that we have talked about at different times throughout the semester. So we're really going to start focusing on that now as we continue to add new concepts and techniques into our tool belt, so we should be a little bit better equipped to talk about these things now. So let's go ahead and get into this. So something that we had said in the past with genetic engineering is that really your kind of peak idea of what you're trying to accomplish is you're trying to make people's lives better. So one example that we talked about is in the case of a disease like sickle cell disease, which we noted last time is caused by a single mutation in the DNA. Well, that's the sort of thing that could potentially be fixed by genetic engineering. Basically, the idea is that you go into the DNA, you change that nucleotide back to what it's supposed to be, you fix the beta globin protein, and then the idea would be that the person would be well again. They wouldn't experience the negative consequences of the disease. Now, we may be a little ways away from making something like that a reality, but genetic engineering as we know it today is going to really refer to the sorts of things that involve producing products like insulin or TPA that make people's lives better. So, like I said, the most common application of genetic engineering is using recombinant DNA technology to make proteins and make all sorts of useful things. Basically, what we've been saying all along, the whole idea of biotechnology in general is to essentially trick cells into doing things that we want them to do, whether it's make something or take something away. We're tricking cells into getting them to do what we want them to do. So genetic engineering is all about manipulating the DNA and making new recombinant organisms that do not exist in nature, with the idea being that these recombinant organisms should do things or provide services that no other naturally occurring organism does. So what you can see in this diagram is the general flowchart to how to actually not only make a recombinant organism, but to extract, say, a protein that organism is going to make for the purposes of marketing it and selling it for biotech purposes. So generally speaking, the steps are going to be, and we'll talk about these as we go along here, generally the steps are ligating the new gene into a vector, so that's going to be a plasmid, which we've talked about several times. So a plasmid, as we said, is just a vector that we can put our gene into, and then we can transform cells with that vector so that they will begin expressing that new gene. So once we have our gene that we want our cells to express, we put it in the vector, we transform cells with that vector. So at this point, we would consider the cell to be recombinant because it is now expressing a gene that it otherwise would not be. So if that gene is some protein, if that gene is instructions for some protein that we really want to make, like insulin or like TPA, we may need to go through a set of steps to induce those cells to make a lot of the protein. In the case of the pGlow lab, the only way we could induce the expression of GFP is if we provided those bacteria with arabinose. Otherwise, they did not make the GFP, as you saw with your own eyes. In a lot of cases with bacteria, genes are controlled by the lac operon, so you actually add a chemical called IPTG, which induces the expression of whatever gene it's under the regulation of. So there are a variety of different ways you can induce protein expression, but it's all really going to depend on what kind of vector your gene is in. But that's a story for another day. So we're also going to talk later on in the chapter about different ways that you have to go about scaling up the production of that particular protein. So even if you induce protein expression, just a couple of cells like the 3 that you're seeing here, they're not really going to make a significant amount of protein on their own. So what we essentially want to do is take those transformed cells and grow them up so that we have trillions upon trillions upon trillions of those cells all working individually to make protein, and then eventually, after we've scaled the whole process up, we can then extract a whole bunch of protein out of a whole bunch of cells. So once we get to the point where we bust open the cells with lysis, we'll have to employ some in vitro methods to start purifying those proteins from the cell lysates. And that will be a topic that we tackle in a later chapter when we talk about chromatography and how to actually purify proteins. So here's another interesting application of recombinant DNA technology, something that we did the very first week of the semester. So the first lab that we did, just to kind of introduce you to what it's like to work in a laboratory setting, nothing too crazy, something kind of fun. You'll recall that we made cheese. So what we did is you were provided with some whole milk, and then you added different chemicals and different reagents to different samples of milk, and you basically assessed how well the milk curdled in response to those agents. So you'll recall that the two agents that curdled the milk the best were Rennin and Chymosin. Rennin is a type of enzyme called a protease, which breaks down proteins, and Rennin is actually a naturally occurring enzyme that is made in the guts of cows. Chymosin, on the other hand, is actually pretty much the exact same thing as Rennin, except it is made in a laboratory. It is actually the product of recombinant DNA technology. And you can actually see the benefit of this because it turns out that synthesizing Rennin from calf stomachs, not synthesizing it, but extracting it and purifying it from calf stomachs is probably kind of a time-consuming and money-consuming effort. So chymosin, if you genetically engineer it, you put the gene for Rennin or chymosin into a vector and you get bacteria to make a whole bunch of it for you. You can make a whole bunch of it very quickly and a lot cheaper than what you have to go through with to get Rennin. So those would be examples of trying to purify proteins from cells. Well, it turns out there are also many cases in which you need to try to isolate DNA from cells, and we'll talk about that in a later section in this chapter. But just to kind of introduce you into the topic, it turns out that a lot of companies, once these methods have been kind of tinkered with and we've worked the kinks out enough, a lot of companies are now selling kits that allow you to very easily and very cheaply isolate either genomic DNA or plasmid DNA from a variety of different cells, whether it's bacteria, yeast, mammalian cells, etc. Extracting the DNA out of cells is not actually that arduous of a task now that we have these kits available to us. But the thing obviously to keep in mind, and something that we talked about in Chapter 7, is that in a lot of applications, if you're going to effectively manipulate the DNA, you need to have it in a relatively pure form. So you need to be aware of things like the 260 to 280 ratio to know how much protein contamination is there, how much RNA contamination is there, things that you need to kind of think about. And then the method of extracting the DNA, I mentioned several different types of cells that you can extract DNA from. The method is going to depend a little bit on where the DNA is coming from and whether it's genomic or plasmid DNA. So we'll talk a little bit about that later, but the first thing that we can say about that is obviously the first step is always going to be the lysis of the cell, meaning that we are going to greatly disrupt the plasma membrane. We're basically going to get the cell to explode so that the membrane pops and all the contents inside that membrane are going to leak out so that we can mess around with them and get what we need to. And then obviously for a lot of cells like bacteria, you need to be aware that a lot of those bacteria have cell walls that surround the membrane and they need to be dealt with as well. Okay, so once the cell has been lysed, the next step, now that we have all the intracellular contents kind of mixed together, we need to separate the DNA, which is what we want, in a relatively pure form. We need to separate the DNA from other contaminating molecules like proteins, RNA, membranes, and things like that. So, as you can imagine, it's not really going to be sufficient to start with just one cell or even a couple of cells. So usually we're going to need a fairly large quantity of cells, usually in the millions to 10 millions, grown in a liquid culture to serve as our starting material for lysis. So it goes without saying, the more starting material you have, the more product that you're going to end up with. It's the same sort of thing that we were talking about in the process of making proteins. You want to scale up the process, get a whole bunch of cells, and that'll maximize maximize the amount of protein, or in this case, DNA that you're going to end up with. So typically what you'll see is we will use the enzyme lysozyme to get around the problem of the cell wall. So lysozyme may in a lot of cases be included in the lysis buffer because it will digest the bacterial cell wall, which is a problem that we knew needed to be dealt with. We can also add a variety of different proteases to break down proteins that we don't want contaminating. Another enzyme called ribonuclease A, or RNase A, can be added to degrade and break down any contaminating RNA molecules. So this seems like kind of a crude method, basically saying, okay, we've got the DNA mixed in with all this other stuff we don't want. Well, let's just break down and destroy all the stuff we don't want, and what we'll be left with is what we do want, which is the DNA. So once the lysis is finished, typically what is done next is that we spin everything down in a centrifuge. So all the high-density stuff, all the cell debris, membranes, things that really heavy proteins, organelles, things that we don't want, the debris is going to be in a pellet at the bottom of the tube, so it'll be kind of stuck down to the bottom of the tube. And then sitting on top of the pellet is going to be what is called the supernatant, which is just the fluid that is on top of the pellet. And usually speaking, the supernatant is going to contain anything that is soluble or low density. So the DNA that we want is going to be in the supernatant in this case. So the supernatant is therefore collected. We already know the pellet doesn't contain anything we want, so all we really need to get out of the tube is the supernatant at this point, which is fairly easy. And then the next step is that we add alcohol to the supernatant, and what this is going to do is that alcohol is actually going to greatly reduce the solubility of the DNA. So the DNA was very soluble in just water before, but once we add that alcohol, the DNA is not really going to want to stay in solution. So at this point, with the DNA falling out of solution, we can either get a stir rod or something like that, and we can actually kind of stick that in the tube and kind of start twisting it so we can spool that long piece of DNA around the stir rod, or we can centrifuge again with the idea that we're going to get two different phases. One that is aqueous, that contains the water and all the things soluble in the water, and then another one that contains just the DNA, and we can get the DNA out of there. So that's the case if you're trying to get genomic DNA, and we understand that genomic DNA is usually very large. We're talking chromosomes that contain millions, if not tens or hundreds of millions of base pairs. So sometimes you don't want the chromosomal DNA. Sometimes you want the plasmid DNA, which is going to be much, much, much smaller. Plasmid DNA is usually going to be no more than 10,000 base pairs. So in that case, the purification procedure is a bit different, and we'll talk about that later. But there are kits for these sorts of things, and they are called mini preps, midi preps, and maxi preps that are just a little bit different and require a little bit different number of cells to start out with. So you may want to pick one of these depending on how much DNA you want and how pure you want that DNA. So one of the big differences is for plasmid preps, the lysis buffer contains sodium hydroxide, which is a very, very, very strong base, the opposite of an acid. It gets neutralized at a later step, but what this really does is it does a good job of making sure that we don't get mixing of the plasmid DNA and the chromosomal DNA. So eventually, the plasmid DNA will get bound to a DNA binding column, and then we can eventually elute it off so that we're just collecting the plasmid DNA and not any chromosomal DNA. Okay, so something that you may want to do after you have extracted DNA out of a cell, whether it's chromosomal DNA or plasmid DNA, is you may want to know whether a very particular DNA sequence is there. So that's obviously a very useful thing to be aware of. So probing for specific DNA molecules that have specific sequences is actually a pretty easy task. So What we're going to take advantage of here that we already know about is the fact that DNA strands bind together with complementarity, meaning that if a sequence of DNA goes A, T, G, C, G, G, C, G, A, T, C, then we know exactly the sequence of the complementary strand that would want to bind to it. So we can actually take advantage of that. So a DNA probe is just a very short piece of DNA, an oligonucleotide, or sometimes colloquially called an oligo, that is not only complementary to the DNA sequence that you are looking to detect, but it also is labeled. It is either radioactive, and we can pick up that radioactivity with a Geiger counter, or it has some type of fluorescent molecule attached to it, which is what you can see in the picture here. So therefore, if we run out a whole bunch of DNA samples in an agarose gel and we get a whole bunch of bands like what you see here, then the bands that have bound to our probe should be fluorescing or should be giving off radioactivity that we can pick up with autoradiography film. So a lot of what we're describing here is germane to something that we talked about before, what is called the southern blot. So we talked in a lot more detail about the western blot. This is the same sort of thing, except with a western blot, we were detecting very specific proteins. Here we are detecting very specific DNA molecules. And you recall, the southern blot is the one that is actually named after someone, Edwin Southern. The western blot is not named after anyone in particular, it's just wordplay. So the southern blot, like I said, involves running out the DNA mixture in an agarose gel like you always do, and then like we saw in the western blot, you are going to transfer those DNA molecules onto some type of DNA binding membrane. So if the membrane is then overlaid with our probe, and then we expose it to X-ray film if it's radioactive, then the radioactivity in the probe will burn a record onto the film, showing the position of the radioactive probe positions in the film. So you can actually kind of see what this looks like on this piece of film right here. And like I said, you'll recall that this is very similar to the method that we used in Western blotting to generate a record of the blot. The major difference between the southern blot and the western blot, obviously, is that it's DNA versus proteins, and then with southern blots, we actually do not need antibodies. We have the probes that will bind to particular DNA sequences. And then it is worth mentioning that there is such a thing as a northern blot, which is almost exactly the same thing as a southern blot, except it is not for DNA molecules, it is actually for RNA molecules. But with RNA molecules, you are still taking advantage of complementary sequences, so the usage of probes is basically exactly the same. Another very important technique that we can go ahead and mention here is the polymerase chain reaction, or PCR. So PCR, the only thing we've really said about it before is that it was invented by a gentleman named Kary Mullis, who actually just passed away within the last year. Nobel Prize winner. This is possibly the most important molecular biology technique that has been invented in the last 100 years. So definitely very cool and definitely worth learning about. So we're not going to talk about this in great detail in this chapter, so I believe a later chapter is going to be where we really start tackling that. But just to give you the general idea of what PCR is it is a technique that allows us, once we have a particular piece of DNA, we can use this technique to copy that DNA and make many, many, many duplicates of it, which is a very cool and very important thing to be able to do. So basically what we are doing is we are synthesizing new DNA, and we can learn from the process of DNA synthesis inside of a cell what sorts of things that need to be present in order for us to get PCR to occur. So a few things that we're going to need in order to do a PCR reaction. We need a small amount of the DNA to be amplified. You really don't need very much of it. So that's why this technique is so cool. If you have a very minute amount of DNA, that's all you need. And then you can use this to make many, many, many copies of it. So you need a little bit of the DNA to be amplified, what we call the template DNA. You're going to need DNA nucleotides, what are called dNTPs. So you need a lot of thymine, you need a lot of cytosine, you need a lot of guanine, and you need a lot of adenine, since we understand those are the building blocks that we use to make DNA. So, for example, if you're going to make a big castle out of Lego blocks, well, you can't build the castle unless you have individual free Lego blocks. So it's the same sort of thing there. We need an enzyme to actually make the DNA for us. We understand already that the enzyme that participates in DNA synthesis is called DNA polymerase, but we actually need DNA polymerase from a very particular organism called Thermus aquaticus, and we'll have more to say about that later. We're going to need all of this to happen in a buffer, particularly a buffer that is suited for that particular enzyme, or rather, the enzyme is suited for the buffer. And then we also need 5' and 3' DNA primers. These are just very short little pieces of DNA that are complementary to both the 5' and the 3' ends of what we are trying to amplify. So it turns out that DNA polymerase can't just sit down on a single strand of DNA and start making another strand. It needs a primer in order to get that whole thing going. And then finally, this machine that you see on the right is called a thermal cycler. What we're eventually going to learn later on in the semester is that PCR cycles through different temperatures. So you go through periods where you're at 55 degrees Celsius, some in which you're at 95 degrees Celsius, and some at which you're 72 degrees Celsius. So getting a PCR reaction to occur is really dependent on being able to go back and forth between those temperatures very, very quickly. So this machine here actually makes that very easy for us because this block right here that sits on top of the samples is a heat block that will rapidly change temperatures to get the temperature of the reactions to be exactly where they need to be. And then, like I said, the enzyme is going to use those primers as basically little bookmarks to know what sequence we want to be amplified. So the 5' primer will sit on one end of the DNA, the 3' will sit on the other, and then everything in between is going to get amplified. So what PCR is taking advantage of is something that we've already talked about before, the idea that DNA replication is what we call semi-conservative. So what that says is that if you start with your original template DNA, which is double-stranded here in blue, if we peel that apart, then each of those single blue strands on the left and on the right can serve as the template for making a new complementary strand. And that's exactly what we're going to do with PCR. So one strand will bind to the 5' primer, and we'll use that to make a complementary strand there. The other strand will bind to the 3' primer, and we'll make a complementary strand there. So the idea is that after every cycle of the PCR, we're going to double the amount of DNA that we make. So basically, if you do this 30 times or even 15 times, every time you double the amount of DNA that you make, so you exponentially increase the amount of DNA that you have on hand, which is especially important if you're starting out with a very small amount of it. So like I said, the thermal cycler is exquisitely important to PCR. So the first reason why you need a thermal cycler is that you need to increase the temperature greatly in order to break these two strands apart up here. Otherwise, they're going to be stuck together and the primer will never be able to bind. So the template DNA has to be heated up to a temperature at which the two strands can no longer be held together. And then every other temperature change is something that we'll talk about later on when we actually discuss the intricate details of how to do a PCR reaction. So we don't need to worry too much about it just yet. So PCR is something that we'll talk about a lot more in Chapter 13, so really we're just kind of covering the very basics here. Alright, so that is going to do it for this first video. Thank you as always for your attention, and I will see you next time, and we will start talking about something that we're already pretty familiar with at this point because of the P-Glow lab that we did about a month ago. Transformation, transfection, and transduction. Basically the idea of getting cells to take up a foreign piece of DNA, and then everything that happens afterwards after they start expressing that gene. So thanks, I will see you next time, and bye-bye.